Hello everyone, I'm Nelson Camilo Rosarios, master student from the University of Illinois, under the supervision of Dr. Hans Stein. I'm going to talk about standardized ileal digestibility of amino acids and concentration of metabolizable energy in three sources of corn fermented protein fed to weanling pigs. This data was presented in the ASA's Midwest meeting in 2024. Soybean meal and corn are the main ingredients used in the swine nutrition. However, have some anti-nutritional factors and don't meet the requirement for some monogastric animals. On the other hand, new co-products from the ethanol industry have been developed with less anti-nutritional factors and less fiber contained that make them more concentrated in protein with good digestibility of amino acids and more concentrated energy as well. However, new technology has been developed in this process to obtain ethanol. Let's see the process to obtain the main co-product that is distilled dry grains with solubles. Let's start with the ground corn. Following cooking process that can ferment it, they use yeast to optimize the distillation to get the main product that is ethanol. But they also get something that is called whole stillage. That whole stillage can be centrifuged and then obtained two other products. The first one, distilled wet grains, and the second one, thin stillage. That thin stillage can be dry by evaporation and then obtain the distilled soluble. Those products can be mixed and dried to obtain the DGS. Now, I would like to show you the new technologies that is called ICM technologies. When I say technologies, it's referenced to new machines that can be added in the process. The first one is thin stillage technology or TH4 that can help to remove the suspended solubles that can be involved in other processes to obtain oil. The remaining is mixed with distilled wet grains, and then we obtain the first product that is corn fermented protein or new corn protein, that is at the commercial name. Additionally, other two technologies have been incorporated in this process. The first one is fiber separation technology, or FST, that help to remove the fiber before the fermentation process by mechanical process. The second one is food optimization technology, or FOT, that can help to use the concentrated protein and optimize the TH4 machine. If these technologies are used together, we can obtain corn fermented protein high in fat content, or advanced 50Y, that is the commercial name. Also, if they added fiber from the other process, they could obtain other product that is called high protein distilled dry grains or advanced 40Y, that is the commercial name. However, these new technologies or new methods to obtain co-products from the ethanol industry can affect the nutritional value of themselves. Therefore, two experiments was conducted to test the hypothesis that the standardized digestibility of amino acid, the apparent total digestibility of gross energy, and the value of metabolizable energy are different among the three sources of corn fermented co-products when it's fed to weanling pigs. Moving to the nutritional value, I would like to show you the products that we use it in the experimental diets. The first one is regular yellow corn, and the other three sources of corn co-products, that they are corn fermented protein, second one, corn fermented protein with high fat, and the last one, high protein distilled dry grains with high fiber content. I will call source number 1, source number 2, and source number 3, respectively, for the corn co-products. And I use the same bar color for this slide and for the rest of the presentation. Let's see the values for gross energy. We observed around 3,800 units for corn. But if we see the co-products, we observe a high concentration in source number 2. That is what we expected because the high concentration of fat, that is the main sources of energy. We will see in a few seconds the fat content that they have. However, if we see the source number 3, the concentration of energy was reduced because the content of fiber that can be affected the energy values, as we observed in previous studies. Moving to the dry matter values, we observe a high concentration in source number 1 than in other two sources. That shows us that probably a longer dry process occurred in this source. But if we see the source number 3, we can see a reduction in dry matter. That is probably, again, for the fiber that was added in the end of the process. They can increase the content of water. Let's see the values of crude protein, fat, and fiber. We observe a high concentration in crude protein in source number 1 and source number 2 compared with source number 3, but higher concentration of fat in source number 2 compared with source number 1. However, if we see the fiber, we found a higher concentration in source number 3 compared with source number 1 or source number 2. That is what we expected because the fiber was added in the end of the process. However, Let me show you more in detail the fiber content. We observe a higher concentration of insoluble dietary fiber, or IDF, that is undigestible for monogastric animals and can affect the digestibility of energy and protein, as has been demonstrated in previous studies. We observe a higher concentration of soluble dietary fiber, or SDF, that can be fermented in the large intestine and be utilized for swine. So let's see the concentration of lysine, methionine, and threonine, that are the limited amino acids. We found higher concentration in source number 2 than in the other two sources. So concentration of lysine, methionine, and threonine, that are the limited amino acids, we found a higher concentration in source number 2 than in the other two sources. What we can observe here is that this corn coproduct is a good source of at least methionine and threonine if we compare with soybean meal or regular corn. Moving to the experimental design for experiment number 1, that was about standardized digestibility of amino acids, was conducted in the Swine Research Center at the University of Illinois, Urbana-Champaign. We used 8 cannulated piglets with the cannula located in the distal ileum and with around 8 kilograms. We used 4 diets, where 3 diets were mixed with each corn coproduct and 1 nitrogen-free diet to calculate the endogenous losses. We fed 3 times maintenance energy, and we used 4 by 4 Latin square with 4 diets and 4 periods, where each period had 7 days, where 5 days were adaptation days, and the following 2 days of ileal collation for 9 hours. Results for this experiment show that the SID of lysine, methionine, threonine, tryptophan, and most of the amino acids were greater in source number 2 and source number 3 compared with source number 1. Something that we did not expect is that the SID of lysine was really low compared with the SID of threonine. This may happen because some of the heat damage occurred during the process. So, lowest SID of lysine could cause increase in mucin losses in the intestine. And then, because the mucin is rich in threonine, it can increase the SID of threonine. Let me show you the design for experiment number 2. That was about a parental total tract digestibility of gross energy and values of metabolizable energy. This experiment was conducted in the same place as experiment number 1, but this time we used individual crates where we allow to collect separate feces and urine samples. We used 32 piglets with around 18 kilograms. They were allowed to 5 diets, where 3 diets contained the corn coproduct, corn, and 1 diet just with corn as the only source of energy. We fed 3 times the maintenance energy, and we used 8 replicates per diet. We have a total of 9 days, where 5 days were adaptation days and 4 days of total collection, where we collect feces and 20% of urine. Let's see the results of a parental total tract digestibility of gross energy. We observe greater values in diets that contain corn or corn and source number 2, but no differences between diets that contain corn and source number 1 or source number 2. However, if we observe the metabolizable energy in diets that contain corn and source number 1, we have a greater values than the diets that contain corn or corn and source number 3. This shows us that adding source number 1, source number 2, you can increase around 125 to 160 units of metabolizable energy in corn diets. Moving to the metabolizable energy in corn and corn fermented co-products, as we expected, we found greater values of metabolizable energy in source number 1 and number 2, comparing to corn or source number 3. That lower content of metabolizable energy in source number 3 maybe is because the higher concentration of fiber that have been demonstrated that can decrease the metabolizable energy value in at least the DGS, that is the closer product that we can compare with these products. Moving to the conclusions, we found that the source number 1, or corn fermented protein, or new coprotein, as a commercial name, has a lower value for its ID of most of the amino acids. This may happen because the high concentration of solubles or heat damage occurring during the process. Heat damage is completely related to the lower SID of lysine or threonine, as we discussed before. Metabolizable energy, we observe greater values in source number 1 and source number 2 compared with source number 3, but no differences between source number 3 or corn. Finally, values of metabolizable energy and SID in these coproducts can be used for diet formulation. However, More research is needed to determine the inclusion rate, and for further research, it's better to determine the reactive lysine to confirmation of the heat damage was occurred. Finally, I want to say thank you to my team, that is Hans Stein Monogastric Laboratory, and the Anderson Company, who funded this project.